Welcome to BioWise with Achal. Now, tomorrow is the examination, and let us have a very quick review of all the important diagrams that could be asked to draw. And there are few diagrams which are required for you to understand certain positioning of the organs. So, let us start with the important diagrams for the biology board examination. The first one, now few diagrams I have made so that it's easy for you to understand. Now here the first diagram is of chromosomes. You have to remember, children, marks of the diagram can be 2 or 3 depending upon the question. And even if it is 2 or 3, see to it that your diagram has minimum 2 labels and maximum it can be up to you. Do not write wrong labels, okay? Write what you know. Fine. So minimum 2 labels compulsory for all the diagrams. Now here if you draw a chromosome, centromere, P arm, Q arm, sister chromatids, you can easily write. Then this is synapses crossing over where you have sister chromatids, chiasma formation. Then you have non-sister chromatids, that is maternal and paternal chromosomes. And finally you have the recombinant chromosome. Right? Easy. Moving to the next is the mitosis phase. Now here what I feel anaphase diagram is little important. Now here I have tried to maintain the chromosome number as 4. Okay. Now in the interphase the chromosomes will be thread-like with the centrioles in position, centrosome in position. As it moves to prophase, you have centrioles moving away from each other to the opposite poles and the chromosomes condense, nuclear membrane disappears. So all these things can be the label. Then metaphase, you have the chromosomes aligned in the equatorial plate in the central line. Chromosomes, spindle fibers, centrioles, centrosomes, centromere. So you can have many labels. There is no problem in labeling the diagrams. Anaphase, try to have a short furrow, a small furrow in the middle, and then anaphase means away. The chromosomes moves away. So have them as it is shown. And finally, the telophase, where you have two daughter cells being formed. And again, reappearance of the nuclear membrane and the centrioles in position, centrosomes in position. Children, see the diagram very carefully. Diagram can be given in diagram-based question. And the next phase, that is, draw the phase which follows this. Draw the phase which precedes this. Precede is the before one, follows is the next one. This is a nucleotide. Nucleotide is what? Phosphate, sugar, and base. Adenine base pairs with thymine by 2 hydrogen bond, and G base pairs with C by 3 hydrogen bonds. It is all DNA is always anti-parallel. So you have the in one chain the sugar is facing up, in the other the sugar is facing down. That is 5' to 3' position. Then here the diagram is of root hair. The first root hair diagram is fully turgid root hair, and the second root hair is a flaccid root hair. The question can be draw the diagram of a root hair placed in a hypertonic solution. Then you have to draw this diagram. Okay, so both the diagrams I have shown. Okay, moving to the next. This is stomata. The guard cells, remember the inner wall has to be thick, the outer wall has to be thin, and then you should have the opening, that is the stoma. The guard cells should have chlorophyll, chloroplast. You should label it. I have not labeled it. Label it chloroplast. Neighboring cells are epidermal cells, cell wall, guard cells. So this is your stomata diagram. Easy? Do not make a flower, please. Huh? Draw the neat label diagram of the stomata. Then this is chloroplast. Chloroplast, the stack of coins, that is thylakoid is the coin, stack of coin is grana. Okay, so this is granum. Stroma is the fluid present. It is a two membrane. Fret is a connection. Thylakoid is a site of light reaction. Stroma is a site of dark reaction. It is the thylakoid whose membrane has got chlorophyll in it, which traps the sunlight. Now these are the blood cells. Erythrocytes, RBC, very important. In many question paper, you must have seen lifespan of RBC 120 days. Then why RBC does not have a nucleus? Why RBC does not have mitochondria? Why RBC does not have endoplasmic reticulum is very important. Granulocytes have granules. Neutrophils, numerous lobed nucleus. Eosinophil, headphone-shaped nucleus. Basophil, bilobed nucleus. Labeling I have shown for one diagram. Whichever diagram is asked, you can draw. Cell membrane, nucleus, granules. For a granulocyte, by the size you can see monocyte is the largest WBC and lymphocyte has got the largest nucleus. Lymphocyte, large nucleus. Monocyte, large cell. Now, neutrophils and monocytes, they are both that is, they perform phagocytosis. So, neutrophils are the first line of defense, you can say, where it is non-specific defense mechanism where neutrophils engulf all the bacteria. So, diapedesis is also basically performed by neutrophils. But monocytes later on becomes macrophages and they help in cleaning the dead and the debris present in your body. So the first attack is done by neutrophils, but prolonged attack and removal of the waste and cleaning is done by monocytes. Both are phagocytotic cells. Then you have arteries, veins, and capillaries. Write in the comment section what is 1, 2, and 3. Okay, 1, that is outermost layer. What is the name given to it? Third one, what is A? A is an artery, B is a vein, and C is capillary. Capillary has single cell, so easy diffusion takes place. But vein and arteries, you can see the structural difference. Question is asked based on structural difference and function. If function is asked, you talk about carrying oxygenated and deoxygenated blood with exceptions. And if structural difference is asked, in presence and absence of valves, wider lumen, thick wall, to talk about that. Write in the comment section what is 1, 2, and 3. Fast. What is 1, 2, and 3? Write it in the comment section. Diapedesis generally do not come for drawing, but you should know what is diapedesis. Oozing out of WBC from the blood capillaries at the site of infection. Now 2 is the WBC and 1 is RBC here. Second one, B is showing phagocytosis. That is engulfing the foreign particles. Then you have the excretory system. The kidney, you see that the right kidney is slightly lower than the left kidney to accommodate liver, right? And then you have ureter, urinary bladder, urethra. Urinary bladder function is temporary storage. So temporary word is very important. Ureter also carries the urine from the renal pelvis or the kidney to the urinary bladder. Okay, and urethra is micturition happens through urethra. Then you have the TS of the kidney and the Malpighian capsule. Always remember in Malpighian capsule, afferent arteriole is wider than the efferent arteriole. Okay, this creates a pressure in the glomerulus for the ultrafiltration to take place. So this diagram is very, very, very important. Kidney, TS of the kidney, you should know how to label it. Okay. The cortex is basically dotted in structure because of the presence of the that is nephrons, Bowman's capsule, and PCT and DCT. And the medulla is all striped because of the presence of loop of Henle and collecting duct. I hope this is clear to you. Then you have the nephron. You can see afferent arteriole is wider than the efferent arteriole. Bowman's capsule, proximal convoluted tubule, distal convoluted tubule, and the loop of Henle. Okay. Ascending spelling, please do the correction. Then you have the neuron. We are drawing neuron 6 standards. Then please, if the diagram is asked, be very specific when you draw. Cyton, pericardion, that is the head part. Axon is the tail. Axon terminal is the last, which is connected to the next dendrites for the transfer of impulse. Okay. Then next one. This is the human eye. Now, human eye diagram. Remember, pupil, iris, cornea, light enters. As the light enters and hits the retina, that point is called the fovea, that is macula lutea, right? And vitreous humor, optic nerve. What is another name for macula lutea? I just gave the hint. You write it in the comment section. What is the other name for the macula lutea? That is yellow spot. What is the other name? Fovea, what? And optic nerve, the place where the optic nerve originates, that is the blind spot. Okay, the muscles which helps in the expansion and contraction of the lens is the ciliary muscles, correct? And this expansion and contraction of lens is called accommodation. And when the pupil constricts and dilates, that is for light adaptation and dark adaptation. Then how is myopia corrected? Myopia is corrected. You can see here in myopia, the image is formed in front of retina. And once you use concave lens, the image is formed on the retina. The diagram can be asked to draw. Then hypermetropia, the image is formed behind the retina, and if you use convex lens, the image is formed on the retina, and the problem is the person cannot read in this case. Then this is your inner ear or the membranous labyrinth with, right here, since middle ear is also shown. So eardrum that is connected to the outer ear, the pinna, the ear, the auditory canal, then the eardrum that is connected to the inner middle ear, which is malleus, incus, and steps, that is the ear ossicles, and finally it is connected to the inner ear or the membranous labyrinth that has got three circular canals at 90 degree each, and with a extension, snail-like extension, that is the vestibule and the cochlea. Then the auditory nerve basically is vestibular nerve and the cochlear nerve that helps in the vestibular nerve, and what it helps in detecting the balance. You know that your ear is responsible for balance. The semicircular canal is for dynamic balance. The vestibule is for static balance. Do not write vestibule. If you write vestibule, write uticulus and sacculus also. Semicircular canal is important word. Uticulus and sacculus is important word. These should be there. If they ask about dynamic balance, write semicircular canal. Ampulla of semicircular canal. If they write ask static balance, then write uticulus and sacculus. Okay? Cochlea has got organ of Cottai that helps in hearing. The sensory cells responsible for hearing is organ of Cottai. They are phonoreceptors. Similarly, photoreceptors are rods and cones. Then the human sperm diagram, the head, the middle piece which has got mitochondria. Then you have a flagellum or the tail, the axial filament, acrosome, and the nucleus is present in the sperm. This is the human ovum, nucleus, the cell, that is the outer membrane. Then you have cytoplasm, inner membrane, zona pellucida, radiata. These are present. This number of cells present around the ovum. Any doubt? Put it in the comment section. Then this is the female reproductive system. Generally, it is not asked to draw, but you should know each and every part. Where are the specific site of fertilization? That is the fallopian tube. Site of implantation, that is uterus. Okay. And yes, endometrium thickens. Ovary is a site of ovulation. 14th day, because of the increase in the luteinizing hormone, ovulation occurs. After ovulation, the ovum travels in the fallopian tube. If sperm is present, then fertilization happens in the fallopian tube. Then the cells multiply and starts rolling to the uterus. Once it reaches the stage of 16 cell stage, it gets implanted on the thick endometrium wall of the uterus. If fertilization does not happen, then this thick endometrium wall shades and it leads to menstrual cycle. Then the male reproductive system, remember the order: seminal vesicle, prostate gland, and bulbourethral gland are in order. Okay. Then you have the testis, through which vas deferens rises. It goes in front of the urinary bladder, right? And they have a common opening, that is urethra. Then this is the embryo growth in uterus, right? You can see the baby growing. Then there is placenta, amniotic fluid, umbilical cord. These all things are very important. Amniotic sac, that is there. Okay. Then you have um, tubectomy and vasectomy. In tubectomy, fallopian duct is cut and ligated so that the fertilization does not happen, the ova does not meet the sperm, and no babies. Similarly, vasectomy mein vas deferens is cut and ligated. This is all. You have mastered the important diagrams of the board examination. All the best, children. Have full faith. You will do well. Okay. Be connected with BioWise. Keep learning. Thank you so much.